Hello and welcome to SAM online class. This is Dr. Subrat Kumar Mohanty. Today we shall discuss about artificial seeds. Artificial seeds are artificially encapsulated somatic embryos or other vegetative parts like shoot buds, auxiliary buds, cell aggregates or any other micropropagules that can be sown as seeds and can be converted into plants under in vitro or in vivo conditions. Artificial seeds are useful for propagation of commercially important crops and for the propagation of elite plant genotypes. This diagram indicates about various components that are included in an artificial seed. This artificial seed has been produced from the somatic embryo. This is the somatic embryo, and somatic embryo is the basic generative component of the artificial seed. And this is the encapsulation matrix. This encapsulation matrix provides protection to the somatic embryo. Because somatic embryo don't have seed coat, and also they don't have endosperm. So to ensure the survival of the somatic embryo, various components like nutrient elements, growth regulators are also included in the encapsulation matrix. In addition to this, other components like antibiotics, antifungals, pesticides are also added to the encapsulation matrix to ensure the survival of the somatic embryo. Essential requirements for the production of artificial seeds. The most important component of the artificial seed is the explant material. The explant material is the basic generative component of an artificial seed. Various explant materials have been used for the production of artificial seed. First is somatic embryo. Somatic embryos are most common propagules that have been used for the production of artificial seeds. And somatic embryos are bipolar structures. They possess both apical meristem and basal meristem, so they can germinate like seeds and form root and shoot simultaneously. Second type of explant material is apical shoot tips. Apical shoot tips, auxiliary shoot buds, microshoots have also been encapsulated in a suitable matrix to produce artificial seeds in some plant species. Other explant material like embryogenic masses and protocorm-like bodies have also been investigated to test their ability to form artificial seeds. In addition to explant, the other components that are included in the production of artificial seeds are nutrients, growth regulator, fungicides, pesticides, microorganisms, for example rhizobia, activated charcoal, antibiotics and a suitable gelling agent. First is nutrient. Nutrients include both organic component and inorganic component. The organic component is the source of carbon. Next is growth regulator. Growth regulator include various phytohormones that promote the development of root and shoots. Next is fungicides. Fungicides prevent fungal contamination, and pesticides protect the artificial seed from different types of pests. Even microorganisms like rhizobia can also be incorporated into the artificial seed. Other component is antibiotics. Various antibiotics are also included in the encapsulation matrix to prevent microbial infection. Activated charcoal can also be added. Activated charcoal improve the vigor and conversion of encapsulated somatic embryo. Activated charcoal break up the alginate and increase the respiration of somatic embryo. It also retains nutrients within hydrogel matrix and release them slowly for the utilization of somatic embryo. Other component is a suitable gelling agent. Among various possibilities, alginate is the most suitable gelling agent for encapsulation of the somatic embryo and other explant material. Types of gelling agents used for encapsulation. Alginate encapsulation is more suitable for the production of artificial seed. Alginate hydrogel is more frequently used as a matrix for artificial seed because of its moderate viscosity, low toxicity to the somatic embryo, low cost, quick gelation and biocompatibility. Alginate enhances capsule formation. The rigidity of the alginate bead protects the encapsulated somatic embryo from mechanical injury. Next, principles and conditions for encapsulation with alginate matrix. The basic principle is that when sodium alginate droplet containing a somatic embryo is dropped into a solution that contains calcium chloride, bead formation occurs. Bead formation takes place due to the exchange of sodium ion and calcium ion. The rigidity of the alginate bead depends upon the number of sodium ion that are exchanged with calcium ion present in the calcium chloride solution. So bead formation occurs due to exchange of sodium ion and calcium ion between sodium alginate and calcium chloride. Types of artificial seeds. Two types of artificial seeds are produced from encapsulated somatic embryos. First category is desiccated artificial seed. Second is hydrated artificial seed. Desiccated artificial seeds are produced from somatic embryos either naked or encapsulated in polyoxyethylene glycol followed by their desiccation. The desiccation tolerance of somatic embryo can be induced by applying a maturation medium of high osmotic pressure. Desiccation tolerance can also be induced by applying sublethal stresses like low temperature and nutrient deprivation. Hydrated artificial seeds. Hydrated artificial seeds are more suitable for the species which are sensitive to desiccation and which are recalcitrant. Hydrated artificial seeds are produced by encapsulating somatic embryos in a suitable hydrogel. This is about types of artificial seeds that are produced from encapsulated somatic embryos. What are the limitations of artificial seeds? There are several limitations. First is limitations in storage due to lack of dormancy. Second limitation is there is synchronic deficiency in somatic embryo development. That means the development of somatic embryos is not synchronous. So, different somatic embryos are in different stages of development. And also there is improper maturation of somatic embryo. There is also a low level of conversion of somatic embryo into plantlets. And finally, there are limitations in production of mature viable somatic embryo. Uses of artificial seeds. Artificial seeds have several uses. First use is artificial seed production is an essential technique for the proliferation of species which are not able to produce seeds. For example, seedless grape and seedless watermelon. Second use is artificial seeds are also useful for mass propagation of elite genotypes. And artificial seeds production through somatic embryo is an important technique which can be used for the propagation of genetically engineered plants and crops. In addition to this, artificial seeds are also useful for the propagation of male and female sterile plants which are used for the production of hybrid seeds. Other benefits are artificial seeds are essential for the production of pathogen-free plants. And also artificial seeds can be directly transferred to the field for subsequent development. And the plants that are obtained from artificial seeds are genetically uniform. Artificial seeds are also necessary for germplasm storage. These are the uses of artificial seeds. Friends, please share, like and subscribe this video, and also please give your comments. See you, bye bye.